applications in things like water purification, sustainability, or biomedical applications. In terms of materials that do have a molecular recognition capture and release function, the certain types of material frameworks that have been used have include metal organic frameworks, covalent organic frameworks, or electrochemical membranes. But since this is a bio-inspired webinar series, I'm here to say that, bio-inspired structures for this specific type of selective capture, release, and reclamation haven't really been explored for materials for this purpose. So so for the next 20 minutes, if you're going to walk away from my talk, I want you to have two key takeaways, and here they are. So the first is that we have synthesized a prototype of this phosphate binding material. And in our characterization, we've confirmed that it can capture phosphate selectively and also release it from multiple cycles of reuse. So I'm going to detail our design of that, the material characterization, and then also the phosphate binding characterization. And then I'm going to highlight some work done by our collaborators using molecular simulations that have helped us figure out some interesting binding mechanisms that we're using for a next generation of material design. So the long-term goal for my work is that we aim to make this a gateway platform that we can optimize and easily alter to be able to reclaim a variety of valuable resources. So that's where we're going to be going. So how specifically are we going to use the protein ability to capture specific capture and bind to specific targets? So the material that we're using is peptide amphiphile micelles. And what that is, here, let's see. I'm going to actually, if you can see my mouse. So a peptide amphiphile is a molecule that has a hydrophobic component. It's usually a fatty acid. And then it's conjugated to a peptide, which is just a short chain of amino acids, or just a shorter version of a protein. And then when you dissolve these molecules in water, they spontaneously self-assemble by shielding the hydrophobic components of the molecule from the water, and then systematically displaying the peptide to the aqueous environment. And this specific material type is interesting because we can utilize, you could call it precise building block components that either the hydrophobic component or specific components of the peptide part, you can tune to control the material design on the macro scale, and also functions that mimic what proteins can do. So we have designed our material from the bottom up, and in order to give you, in order to let you see what I've done, I'm going to share some background useful material for you to interact with. So how are we able to, have these precise building blocks? And we do that through precision synthesis techniques. And specifically, we use solid phase peptide synthesis. And the way that works is, you have a solid support, and then you can sequentially couple specific amino acids and basically obtain a precise control of the sequence that you want, and also a monodispersed product. And this control of amino acid sequence is critical in developing specific binding materials. So the way that we're designing this to have material functions and also protein mimicking functions, we need to have specific sequence techniques. And so we can precisely synthesize, but we can also tune how these materials self-assemble. And we can do that via two different factors. One is more of a steric factor, and you can use the so-called packing parameter. And so this is a ratio of the volume of the tail component to the area of the head group. And according to what ratio that is, you can tune it to self-assemble into spherical micelles, worm-like micelles, or other shapes such as vesicles or bilayers. And depending on what application you want to use it for, you can control that factor. And then also for peptide amphiphiles, we can control the head group interactions of the peptides to one another within the self-assembled structure. So you can control that via electrostatics, or also hydrogen bonding that form specific beta sheets that can push the assemblies towards worm-like micelles. And so both of these factors we incorporate into our design of our structures. So the last background slide before I get into my work is that we can also incorporate specific biomimetic functionality for the peptide amphiphiles as well. And so these consist of one, secondary structure control. So just like I said, we have the building block components that we can design in, and some of these building blocks we can specifically put in to help it self-assemble or help the peptide within the corona adopt a secondary structure, such as alpha helices or beta sheets. And we know that not just primary structure of proteins, but certainly secondary structure is critical in binding. And then second, this material platform has widely been used for already specific targeted binding and recognition. And it's used for targeted drug delivery and also cell adhesion are two common uses of this material platform. So so we do that by synthesizing pre-identified binding sequences into the peptide head group. And then lastly, this material platform can easily incorporate multiple functionalities within the same structure by simply dissolving different peptide amphiphile molecules into the same aqueous solution, and they'll self-assemble, and each of the constructs will have, multiple functional groups depending on what your peptide sequence is. So that it makes it just a really versatile and easy to work with material. Okay, so hopefully that was helpful background for you to know where I'm going now for my design. So just to sum up what I said, so peptide amphiphiles possess really important structural and binding advantages, but so far they've only been applied to target biological macromolecules for biomedical applications, such as the drug delivery that I spoke about. Second, here we are pivoting from that specific application and design to use this platform as a basis for targeting and harvesting small molecules and ions within the micelle corona. So as opposed to being the specific target to adhere to something else, suddenly now we're using it to be an absorbent or harvesting type material based on this selective binding of proteins. And so how is this actually going to work? So our aim of our project is to create a dense suspension of entangled worm-like micelles, and you can see it in this cartoon on the right, that are able to capture and release phosphate in the head group, so along the strands of this self-assembled construct, and then release it according to a pH trigger. And this could have interesting applications. One specifically for phosphate is we could use it to reclaim it as a fertilizer, especially since there's a lot of agricultural runoff that contains excess phosphate and causes eutrophication. And then there's also biomedical applications for it as well. So that's the background. And so now I'm going to share first the design of our materials, then the material characterization, and then also the phosphate binding abilities. Okay, so how did we design our material? So as I said, we are utilizing a protein-derived phosphate binding sequence that has been well studied and characterized. And there's a specific amino acid sequence known as the P loop that has been identified in phosphate binding proteins that usually binds to the phosphate of ATP. And this binding mechanism occurs through a nested cavity formation that's depicted in this cartoon here, and also through hydrogen bonding in the backbone, and then a lysine side chain that comes around and stabilizes the phosphate. And so this specific hexapeptide sequence that we chose, SGAGKT, has already been synthesized synthetically and shown to bind phosphate in a pH-dependent manner. So that specific sequence we are then taking to add on to, here's the peptide amplifier molecule that we've designed and characterized. So this is the phosphate binding sequence that we've incorporated on the periphery of the peptide amplifier molecule, so that you can imagine that it's self-assembling into these worm-like constructs. The functional binding part is extended into the aqueous environment. And then the blue dotted spot here, if you remember from the material design, if you incorporate specific glycine spacers, that helps it, forces it to adopt a worm-like micelle formation because of hydrogen bonding that occurs between beta sheets of the aligned peptide amplifier molecules. And then we also have incorporated this fatty acid tail, which is what drives the hydrophobic self-assembly. So this is the molecule that we've used, and we've denoted it C16GGG hex for the hexapeptide. And so this is what we synthesized, and we purified this using the solid phase peptide synthesis technique that I discussed previously. Okay, so we designed it, and then we synthesized it, and the first step was to characterize, okay, how does it self-assemble? Does it self-assemble? And then if so, what types of assemblies and what are the length scales? So indeed, it does self-assemble, and we've these are negative stain TEM images of the self-assembled constructs. And at pH 6 and pH 11, we see that it self-assembles into extended worm-like micelles, which is great. That's the construct that we want to form this, dense entangled network for our application. But what is interesting here is that, at pH 6, we see it, basically be evenly dispersed, but then at pH 11, we see this clumping effect of the micelles. And we believe that is due to there's a lysine that's involved in the peptide head group, and that gets deprotonated above its pKa of 10.5. And so with that, likely solubility is decreased. And so then they prefer to to clump together. So I'll have you remember this specific phenomenon for something later that we figured out in terms of how it affects our binding. But yeah, overall, conclusion from this slide is that this system is well suited to form a dense suspension to retain and release phosphate, which is what we want. Okay, so having characterized the material properties that we care about, we can move to looking first at does it bind phosphate, and if so, what are the conditions? And indeed, it does. So this plot on the left is the percentage of phosphate bound versus pH of the conditions of binding. And it's this plot specifically is a ratio of 5 to 1 of the peptide amphiphils to phosphate in solution. And so we see 100% binding is achieved at around a pH 6, and then binding is decreased as pH either increases or decreases. And we see pH 2 and pH 11 are the potential release conditions if we're thinking about making this a capture and release material. So and then the plot on the right, this is at pH 6, the maximum binding. We see that as we increase the amount of peptide amphiphil units present, we're able to, increase the amount of phosphate that's bound, which makes sense. But so when we have equal molar peptide amphiphil binding unit to phosphate, only about half of the phosphate is bound. And then as we increase that roughly about to 3, we're able to basically achieve 100% binding. And I should say that these experiments were primarily conducted by 2 undergraduates that I've had the pleasure of working with, and they did a great job. So Tess and Gabby. Okay, so we checked that it binds to phosphate, which is great. And so next we wanted to check and see, can it release it? So there's a lot of absorbent materials out there that can bind specifically to targets, but then they're unable to release it. And that's one of the main goals of this project, is to be able to recycle what we want. So we checked, we did sequential pH adjustments of the material, and we overall determined that it is able to capture and then re-release the phosphate, which is great. So this plot here, we have percent phosphate bound again on the y-axis, and then the number of cycles that we sequentially adjust the pH from 6 to 11 to 6 to 11 to 6, or 6 to 2 to 6 to 2 to 6 to 2. And what you can notice here is that the pH 11 curve, which is the blue dotted one here, after about 3 cycles, it is only able to bind to around 50% of the phosphate, which we think is probably related to the clumping phenomenon that we saw. But for pH 2, we see that the binding does decrease over time, which shows that there's room for optimization here. But overall, we capture that this phenomena of sequential capture and release is possible for our network. So the last experimental thing that we wanted to check is it selective? So great that it binds phosphate, but is it just doing it because we have a positive charge and it's targeting a negatively charged anion? So the 2 competing ions that we chose are nitrate and nitrite, and we did that specifically for the application of fertilizers. So fertilizers largely consist of phosphorus and nitrogen molecules. And so we want to be able to see, can it extract phosphate out of a nitrate-rich environment? And in these experiments, we see that it is selective of phosphate over nitrate and nitrite. So again, we have the percent of anion bound on the y-axis, and then 3 different binding conditions on the x-axis with, we did equal molar peptide amphiphil to each of the anions, and then we increased the amount of binding units, or the amount of peptide amphiphils, while keeping the equal molar concentrations of the anions the same. And at this 1-to-1, the equal molar concentration, we see that essentially it is very selective, and it binds to phosphate about the same that we saw when there was no competing anions present. And then as we increase the amount of peptide amphiphils present, we do see some nitrate and nitrite begin to bind. So overall, we are excited to see that there is selectivity, and this confirms that it likely is employing what the protein-inspired binding mechanism that we want to design into this material. But there's definitely room for optimization as well. And so we have some ideas about what to do next for future rounds of design. Okay, so going back to the key takeaways for my talk. So I just presented the basic information that we have synthesized and characterized this phosphate binding material, and confirmed that it can bind phosphate, does so selectively, and it can do it for multiple cycles. And then for the remaining few minutes of my talk, I'm going to talk about some interesting insights that we gleaned from molecular simulations that help inform future rounds of design. So Yeah, that's where we're going to go here. So to recall, so this is the phosphate binding profile of our material, where we saw that phosphate was bound at a maximum of pH 6 and minimum at pH 11 and pH 2. But that is actually quite different than the pH binding profile of the non-assembled binding peptide. So when it's just the hexapeptide, that was previously synthesized and shown to bind phosphate. This is a pretty confusing graphic, but if you look with me at this dark green line, that's actually how they characterize. So L is the ligand or the peptide binding to this phosphate, and we see that it binds most maximally at 10.5, and then it also binds to phosphate here, but then they propose that there is no binding that occurs at pH 6 or below. And so this was from the original paper characterizing this in 2012, and then there's actually been 2 studies since then that have confirmed this result, that their maximum binding is achieved at high pH and no binding is achieved below pH 6. So our question is, so why is our system noticeably different? We see maximum binding at pH 6, they see maximum binding at pH 10.5, we see no binding at pH 10.5, and they see no binding below pH 6. So some ways we thought about, why could this be the case? So is it on the molecular level? So did the tail and the, added amino acid spacers, did that somehow change the binding properties? Or did the self-assembled construct induce the change? So to answer this question, we turn to molecular simulations. So these were done by our collaborators in the DePablo group, and specifically by Chuting. And so she did great work here. So she set up these simulations, and her work captured this pH shift as well. So when she simulated just the single peptide amphiphile, not in the assembled construct, she also saw that binding occurred at a maximum around pH 11. So in this figure A, pH 11, these are the top 5 most common conformations, and all of them show phosphate binding to the peptide amphiphile. And then at pH 6, of the top 5 that were most commonly present, only one of them showed binding. And then she did a free energy mapping of it. And again, you see a free energy minimum here for the pH 11 condition, showing that, it's stabilized at a certain distance, the phosphate from the binding unit. And then at pH 6, it's stabilized somewhere out in the environment, showing that phosphate is not bound. So she was able to, confirm the previous results of the single peptide experiments. But then when she simulated a self-assembled system, she captured what we saw, that binding then suddenly was able to occur at pH 6 and did not occur at pH 11. So we can ask, again, why does binding not occur at pH 11, and why does binding occur at pH 6? So just last couple slides before I'm done. So at pH 11, what she saw was that the micelles essentially get squeezed, and she analyzed this via a solvent accessible surface area analysis, where at pH 6, as it sounds like what it sounds like, that you can analyze the surface area of the micelle that is accessible to solvent, and at pH 6 it's larger, and at pH 11 it's smaller, showing that the micelles shrink in size. And as the surface area decreases, there's a decrease in access to binding sites. So phosphate can no longer penetrate into this super dense micelle corona and adopt the conformation that it needs to bind phosphate. So that answers the first question. And then the second question is, why is phosphate bound at pH 6 when it wasn't bound before? And what we see here is a pretty interesting phenomena that I am using to design and test the next generation of things, is that we see multi-chain binding employed within the micelle corona. And so again, obviously this would not be possible for just a single chain peptide, but within the self-assembled construct, where we have all of these peptides that are very close, suddenly multiple chains are then being used to stabilize the phosphate. And one could say that this is actually reminiscent of tertiary binding mechanisms of proteins. So we know, obviously, that the specific amino acid sequence is important, the secondary structure is important, but even probably more so than both of those are the tertiary and quaternary structures of proteins to be able to bind to their specific targets. And what we see here is that our system has a lot of potential to actually be able to incorporate and design in more specifically tertiary binding constructs. And again, we see this confirmed with the experimental data. So if you remember, we only saw 100% binding with around a 3-to-1 ratio. So that's just, again, confirming that multiple peptide amphiphile units are required to bind a single phosphate in this current setup. So in terms of my future work, I am going to utilize the mixed micelle design scheme that I talked about at the beginning to be able to enhance the selectivity and hopefully more closely mimic this protein binding functionality. So that's the end of my talk. To sum it up, 3 main points. The first one is just very generally, if you're going to walk away with something, just to remember that peptide amphiphile micelles are a new interesting material platform that can exploit the binding ability of proteins in a way that's practically usable, so we can actually reclaim the phosphate rather than just having it be in solution. Second, this material bound phosphate 100%, was selective, and also performed multiple cycles of reuse. And then third, the molecular simulations captured this interesting binding shift from the maximum binding at high pH to pH 6 through the squeezing of the micelle corona at high pH, and then the multi-chain binding at pH 6. So definitely want to thank all of my group members and collaborators here and supporters, and I would be happy to answer any questions.